the next would be about the neurology and the clinical implications as such. Before going into the diseases, a bit very brief about the evolution of the nervous system. See, in the protozoal animals, the earliest animals of the world, that's single cell animals, the system of receiving the sensation and receiving and having a reaction to the stimulus is a basic nature of the life. See, any one of the definition of the life is the capacity to receive the stimulus and react is one of the important character of the life, a living being, and a unicellular organism had a system of reacting based upon the localized chemicals or the hormones present in the cells as well as the cell membrane capacity. But gradually, when the evolution occurred, when the multicellular organism developed, there was a gradual development of some specified cells which would be capable of receiving the sensations and then reacting as such. So that's how it's evolved. Like in the lower animal, that's in the unicellular organism, there was no separate system. It was only the hormones or these enzymes which are capable of reacting. Whereas as the multicellular organisms developed, gradually there was some evolution of a separate system, and the cells which would be capable of reacting to the stimulus were developed, and they are specialized. And what we identify as the structures of the nerves, they were seen after the development of the maybe the Giardia species, like these fish species. From that time, from that level onwards, a specialized system was seen. And of course, it's considered that the human nervous system is most evolved. And among the animals, the brain is the major center. That initially, again, a bit more of that. In the species like Hydra, the lower species, every cell was behaving like a sensory system, and then there would be a pathway. But later on, there was a development, and of course, nerve cells were localized in the core area of the body, and there was no such central nervous system. But in the major later on, from the animals of fish and onwards, there was something like a central nervous system, which was located in the head area, and it's also evolved. Like from the mouse to human being, there is a gradual evolution in the size and functions of the brain. That's in brief about that. Now, coming to the human brain and human nervous system, the functioning unit of a nervous system is called as a neuron or the nerve cell. And the nerve cell, a general standard pattern of the nerve cell would be, it would have an axon and then a nerve cell or neuron as such, then dendrites, the other end. Now, the important character of these would be the axon is the connecting point, and it can carry the stimulus from one area to the other, and it's a living cell. Nerve cell is a living cell. Of course, there are some more patterns of the nerve cells. This is a very standard pattern. Depending upon the shape of the nerve cells, they are called as unipolar when there is this axon or the neuron cells at the tip of the nerve cell, and axon and dendrites, the branches which can receive the sensation from the other side. Bipolar is where you have the dendrites on both the sides as such. Then pseudo unipolar, where there is again a sort of nerve cell or neuron in the middle, and then it can carry the sensation from only one direction. Multipolar is where there could be multiple directions as such, the multiple dendrites on both the sides, and hence the impulses can reach to both the areas. Again, another way of classification of these nerves based on their characters are non-myelinated and myelinated. Nerve cells may have a layer of myelin sheath with some node-like structures, which we call as nodes of Ranvier, and that those nodes would be helping in conduction of the impulses. Of course, more evolved would be classification of the nerves in the human beings would be based upon their length and then the speed of conduction as a conduction velocity and their size as such, and whether there is a myelination or not based upon that, nerve cells also are classified into sensory nerve cells are classified as A, B, C, and in the A again you have A, alpha, beta, gamma, and delta. Then B and C, which are among them the C type of nerve fibers are non-myelinated, and they are conducting slowest, rather they conduct the impulses very slow. The passage of the impulses is slower. In myelinated nerve fibers, the passage of the impulses are faster, and again that conduction of type of conduction is called as saltatory conduction of impulses, where the impulses would jump from one area to the other. And alpha motor nerves are considered to be the longest or the largest as such, and they carry the impulses very rapid, and they are mainly motor neurons. And beta neurons, they carry actually the sensory sensations like touch and pressure. Gamma neurons are again, they are motor neurons present in the muscle fibers. We will just have some more discussion about these issues. And delta neurons carry the touch, pressure, temperature, and acute pain. And the beta nerves are preganglionic autonomic nerves. Now, what are those preganglionic autonomic nerves and their functions? We will just continue further when we go into the subject further. We will discuss more about that. And C nerve fibers, the slowest nerve fibers, they are the ones which carry the smell sensation, and they also are postganglionic nerve fibers, and they carry the pain sensation slowly. That's, see, once there is an injury, acute pain, it's carried by either the beta nerves or maybe the delta nerves, whereas the gradual pain, the pain which persists for a longer duration, is carried by the C nerves. Sensory only, there are a few nerves which are only sensory, which do not carry the motor functions, where the direction is unidirectional, the direction of impulses are unidirectional. They are categorized as 1A, B, and 2, 3, 4 as such, and there again they depend upon the muscle spindle nerves which carry the sensation of the tension in the muscles. Golgi tendon organs, again we will have some more discussion about these issues. Then muscle spindles again, so 1, A, B, and 2, these are mainly located in the muscles, which help in measuring the tone of the muscles and maintenance of the muscle activity, or receiving the sensations about the muscle activity to the hair center. Then the third type is about carrying the touch, pressure, and fast pain, and the fourth is pain and temperature and smell sensations are carried by the fourth type of nerves, which are again slowest. Now that's about the different speed as such. Now when I said about the action, this conduction of the impulses is produced due to an action potential. Any cell in a living condition would have a membrane potential in the sense there will be a potential difference across the cell membrane. It will be having more positive charges on the exterior and negative charges on the interior as such. But all the cells may not have an action potential. Those cells which get activated due to the stimulus, any peripheral stimulus, where there will be a change in this potential difference from once it was positive and it becomes negative after a stimulus, we call such cells as excitable cells. Among the excitable cells in the body, nerves and the muscles, these are the two important excitable cells where there can be an action potential. So whenever there is a stimulus to the nerve of any sort, now stimulus could be of any sort, there will be an action potential formed, in the sense temporarily there will be a exchange of the ions, and temporarily one zone of the nerve becomes positively charged inside and negatively charged outside. There will be reversal, and this is what we call as the action potential. Now once the action potential forms at one zone of the nerve, it tends to move towards the other end of the nerve. It's conducted. The action potential tends to conduct it, because nerve is a good conductor. As the electricity passes in a wire, similarly there will be a passage of these impulses. Now gradually there will be a reversal of the same, that potential differences from positive to negative and negative to positive across the cell membrane, and that's how it reaches from one end to the other. So that's how the whole thing becomes actually, the impulse reaches from one end of the nerve to the other. But as the impulse passes through, the area which was having action potential, or which had that depolarization, where the polarity was altered, it gets depolarized and the nerve cell would be ready for another impulse as such. So there can be a series of impulses passing through. As the impulses passes through in a demyelinated nerve fiber, the passage would be by a continuous exchange of, or continuous change in the environment, that polarization of the cell membrane continuously. Whereas in a myelin sheath, these changes would occur at these nodes of Ranvier. They say once at here, then the next change will be at the nodes of Ranvier, again the next nodes of Ranvier, and because of this kind of a movement, it's called as saltatory movement, and this occurs rapidly. So the speed of conduction, velocity of conduction can be changed based upon the nature of the cell. Of course, it also is depending upon the other factors like the status of enzymes, temperature, and so on. Multiple factors would affect the velocity of a nerve, velocity of a conduction in a nerve. But structurally, from the basic point of view, it's whether the nerve is myelinated or demyelinated, non-myelinated. That would be the factor which decides about the speed of conduction. Now, need for these different speeds of conduction would be understood when we go further into the functions of the central nervous system as such. Graphically, the speed is represented as such. In case of myelinated nerve fibers, the speed is vertical. It reaches the impulses reach a high speed velocity very rapidly, whereas in a non-myelinated nerve fibers, they reach to the target area comparatively slowly. Now, whenever there is an action potential occurs, that action potential occurs when there is a stimulus. Now, for an action potential to be generated, the intensity of the stimulus is one of the important factor. If the intensity of the stimulus reaches to up to a certain level, then only the action potential will be found, and that level is called as a threshold level. So threshold level is the minimum intensity of the impulse which is required for producing an action potential in a nerve. Now, this is also called as the excitability of the nerves. Excitability of the nerves would be varying again basically based upon the same structure factors like the classification alpha, beta, so on. C nerve fibers are comparatively less excitable, whereas alpha nerve fibers are more excitable, in the sense the threshold limit of the alpha nerve fibers are lower, and the threshold nerve of C nerve fibers are more. It needs more intensity or higher intense stimulus there. Or even otherwise, even among the nerves, even alpha motor nerves also, depending upon the location, the excitability can vary as such. Excitability also can be varied due to the other factors like the environmental factors. Then there is another important issue. If there is a sub-threshold stimulus, stimulus which is less than the threshold limit, that makes the cells more excitable, but it doesn't produce a action potential. If there is a continuous low intensity impulses which do not reach to the threshold limit, they can summate and the excitability can increase. So if there is a stimulus which is lesser than that of the threshold of a nerve, a single stimulus will not produce the action potential, but if there is a series of such low intensity stimuli, that can result in an action potential. This is what called as a summation. Now, once the action potential occurs, there is a sudden change in the electronic environment, the potential difference, then it becomes negative outside and positive inside, and this is a very short duration, momentary issue. The time taken for this action potential to retain also be varying from nerve to nerve. The nerves which are some slow conducting nerve fibers like C nerve fibers, they have a longer active period, the period where the duration of the potential difference remains, whereas fast conducting nerves would have a shorter duration. The spike, the shape of this spike would be changing. See, in a faster nerve conduction, this spike would be shorter, or the width of the spike would be shorter, whereas in a slow conducting nerve fibers, the width of the fiber would be longer as such. Now, so time taken for repolarization also is one of the factor which can vary, and this again can affect the functions of the nerves, and these also are predetermined for each variety of the nerves, and of course they also can change in the environment, change due to the environmental factors. Now once the nerve fibers have got repolarized, because the repolarization is a temporary event where there is influx and efflux of the ions, but still the environment may not be exactly normal, there would be a situation where further stimulus at that time, the further stimulus may not be able to produce an action potential, even if it is above the threshold period, level, and that period is called as refractory period, or that's also the resting status. It takes some more time, and this period also, refractory period also, would vary from nerve to nerve, type of the nerve to type of the nerve as such, and that decides like whether another stimulus can produce the action potential or not, if it is produced within, if it is reaching within a short duration or not. Suppose if there is a continuous action potentials in a nerve, then gradually the refractory period increases, and this is what we call as the tolerance later. See, suppose if there is a continuous action potentials, a nerve is stimulated continuously, due to that the environmental changes in the atmosphere of the nerves, like maybe the enzymes and the chemicals in the nerves, the nerve cells become less excitable, and hence the refractory period can become longer. So refractory period also would be deciding factor about the excitability. So ultimately, whether a nerve can get stimulated due to a specified stimulus or not, and the how much of the whether which intensity of the stimulus would require to produce an action potential, and how far these impulses would reaching, or how much time is taken for the impulses to reach, is based upon these electronic characters of the cells, nerves as such, like the threshold limit, then the time, depolarization time, repolarization time, and refractory period. These are the factors which decide. But there is another phenomenon called as all or none phenomenon. That all or none phenomenon is once the nerves have a property, if there is a supra-threshold stimulus, a threshold stimulus above the stimulus above the threshold limit, the total intensity of the action potential formed is the same. It doesn't change. So it's not proportionate to the intensity of the stimulus. If the stimulus is stronger or weaker, if it is above the threshold limit, the extent of action potential would remain the same. It's not depending upon the intensity of the stimulus. This is a very important factor, and this phenomenon is called as all or none phenomenon. All or none phenomenon in the sense, if there is a stimulus, it produces the same intensity of changes in the nerve cell action potential, or there is no change in the action potential if the stimulus is less than the threshold limit. So, the severity of the stimulus is not measured by the intensity of the action potential, but it's measured by the other means. We will come to that part later on. So that's about one of the important aspects of the physiology of the nerves. Then of course the next part is, once the one nerve is stimulated, the nerve would be connected to another nerve through a synapse. Synapse is a one junction where two nerves are interconnected together. So once the impulse is generated in one nerve cell, that needs to be spread to the other nerve cell, and this is what we call as a synaptic conduction in the synapse. That conduction in the synapse is purely a chemical. That's once this action potential is formed in the one nerve, and the action potential reaches to the synaptic end of the first nerve, which we call as a presynaptic nerve, and then there is a connection between the two nerves, which is called as the synapse at the dendrite level, and there will be some chemicals discharged, and these chemicals discharged are called as the neurotransmitters. They would be of different type of neurotransmitters. I will just have a brief outlook about these neurotransmitters also, and depending upon their quality and quantity, the next nerve would be either stimulated or may not be stimulated. So and if there is this chemical stimulus would be reaching to the next nerve, and that chemical stimulus will produce another action potential in this nerve, and this is how a stimulus from one zone can reach to the another zone in the body, like from periphery to the brain. So when somebody touches, the touch sensation is received, producing a stimulus in the peripheral nerve, and then the impulse reaches to one nerve, the first set of the nerve. Then in the next set of the nerve, it gets excited or maybe not excited, depending upon the presence of these neurotransmitters, and it may be the same would be converted into an action potential in the next nerve, and that action potential reaches to the next, and so on. If there is a series of nerves, it crosses through the series of the nerves and reaches to the target area. These chemicals are called as the neurotransmitters as such, and they are of different varieties. The most common neurotransmitters which are seen or which are present in the human body, they are classified into 4 groups: amino acids, peptides and biogenic amines, and acetylcholine. Acetylcholine is present almost in all neuromuscular junctions, as well as in parasympathetic nerve fibers and sympathetic ganglionic areas, and they produce excitability. Acetylcholine excites the next nerve cell, and it's synthesized within the nerve cell, and then it gets metabolized there itself. Then comes the biogenic amines. Among the biogenic amines, norepinephrine, dopamine and serotonin, which also are norepinephrine and dopamine are called as catecholamines. They are primarily secreted in the sympathetic nervous system, and they too produce excitability or excited nerve cells. Then there are some more like the vasopressin, somatostatin, neurotensin, which are having specialized function at the hypothalamus. We will have more detailed issue about the discussion about these activities when we go into certain diseases of the hypothalamus later on. Similarly, many of those neurotransmitters in the central nervous system, like aspartic acid, glycine, glutamic acid and GABA, gamma-aminobutyric acid, these also would have their specialized functions. We will have some more detail about these issues when we go into the diseases as such. Important is some of them are excitatory, some of them are inhibitory. Excitatory in the sense some may increase the excitability of the nerve cell, next nerve cell, and hence there could be an action potential produced. Some of them may alter the excitability of the next nerve cell by reducing the excitability. Now, excitability is reduced by or increased by varying the factors like the refractory period, the threshold limit, and so on, the depolarization time, and so on. These can be varied by these chemicals, and hence the next nerve either could be inhibited, that there may not be a action potential, or rather capacity of that nerve cell to produce an action potential could be reduced. It's something like a brake, and excitatory are something like an accelerator. Now that's all about the very brief about the neurotransmitters. Now more of that neurotransmitter discussion, we will have discussion more about that. Importance of these neurotransmitters are also important. Importance will be known when we go into the pharmacology of the drugs related to the nervous system. Some of these drugs can either inhibit this conduction of the impulses and formation of these neurotransmitters, or they may be changing the excitability of the nerves, and some of them may be increasing these. And about this pharmacology, we will have somewhat more discussion when it comes to, because instead of going into that detail now itself, I think we will reserve this for the discussion when it comes to the issue. The neurotransmitters, again the same neurotransmitters, again their structures. Catecholamines, they have a basic structure of a catechol ring and then a aliphatic chain, whereas the others, their structures also are different. Of course, endorphins also are one of the neurotransmitters which are present in the brain, which can produce a feeling of happiness, sensation of happiness. And so there is some more than simply the functions and action potential, but that action potentials produced through endorphins, they can produce a feeling of happiness. Sensations also can be changed as such. So that's about some more of the neurotransmitters. Much more detail of the neurotransmitters, we will come to that later on. Now, of course, this is a point about the excitability of the nerve cells. Now, excitability of the nerve cells, I have referred to that. The basic issue of the minimum stimulus and the duration of the stimulus. Another factor is if there is a low intensity stimulus less than the threshold limit, if the same persists for a longer duration, then it can produce an exact this action potential, and this level is called as chronaxy level. Time duration is called as chronaxy level, and similarly amplitude, the intensity of the stimulus produced also is called as the Rio base. So chronaxy and Rio base, these are the two factors which decide about the excitability of the nerves, and this will have an impact on the amount of the neurotransmitter secreted at the synapse also, and hence the next impulse which passes through the next nerve would also be regulated. Now, in the synapses again, there could be a summation. The synapses, they may be arranged in such a way that if there is more than one synapse, like say if the two dendrites are connected to one neuron, and if both the dendrites, they would produce a stimulus in the presynaptic area, they would produce a stimulus which is less than the threshold limit. But because there are more than one or two or three of these stimuli, they can summate, and there could be an action potential in the nerves. Next nerve. See, in the presynaptic nerve ending, the stimulus produced or the neurotransmitter secreted could be less than the threshold level. But if there are more than two or three such connections together, three dendrites connected together, then there could be a summation, and then action potential can be formed in the next nerve. Or there could be a temporal summation. This is called as spatial summation, where multiple nerve endings, they are connected together. Then there is a temporal stimulation, is if the same one nerve ending synapse which produces a subthreshold stimulus, but the subthreshold stimulus is continuously administered, continuously given, then that also can produce a summation that's called as a temporal summation. Now, that also can be inhibitory. Like if the neurotransmitter secreted is inhibitory, the excitability of the next nerve cell would be reduced further, and the threshold limit would remain the same, but the excitability of the nerve cell can be reduced. This a stimulus can reach to the distal area with varying intensities and varying distributions. Like now, or if there is a multisynaptic level, there can be at one stage there could be a inhibition, another stage there could be a stimulus, and this is what we call as a differentiation. Or there could be the other way, there could be a negative stimulus or inhibition at both the endings, then it's called disinhibition. If it is only excitatory, it will be positive, that's facilitation, and if it is negative or inhibitory, then it's inhibition. Spatial summation is, as I told you, multiple dendrites connecting to a one neuron, and then it can be producing the same. Temporal summation is a single neuron, but where there will be multiple stimuli which can affect the next impulse. So these issues, of course, they are not very complex issues, simple issue, but because of the nomenclature looks like complex as such, but these have to be properly understood. Like a stimulus which is below the threshold limit also can produce an action potential, and it can reach to that area at different manners. So there could be multiple varieties of the ultimate action potential produced due to a simple stimulus in a body, and this can explain many of the complex neurological functions as such. Same temporal summation and the spatial summation, another better diagram which gives the same picture, like which clarifies the same issue. When there are multiple impulses from different organs, different dendrites to a single nerve cell, there could be this excitability as such, whereas postsynaptic potentials would be enhanced as such. Now, the other way, another is nerves, as I told you, they are specified to carry the specific sensations. Now, sensations are carried by the specialized structures at the nerve endings. So, like some of these nerve endings, they are modified in such a way that they can carry only a specified sensation. Like in the, of course, in the diagram we have not added that the optic nerve, it's carrying the only vision, light sensation. Olfactory nerve, it carries the smell sensation. Among the touch sensations over the surface of the body, which carry the touch, temperature and pressure, etc., the free nerve endings, they carry the touch sensation, and a high touch sensation can be considered as a pressure, the pain sensation. Then expanded tip receptors, the receptors, nerve endings which would have a fine nerve endings at the skin, they carry the touch sensation. Expanded tip receptors, they would carry somewhat pressure sensation, moderate pressure sensation, whereas the Pacinian corpuscles, they carry high pressure sensation. Like, see, what I mean by that is, when somebody touches, fine touch, that produces a stimulus in the free nerve endings present in the skin area. Suppose the number of free nerve endings at some areas of the skin are lesser, then that intensity of the fine touch sensation felt by a person would be lesser, whereas or less intense. Whereas those areas of the body which have higher sensations, higher number of nerve endings, the intensity of the touch sensation felt to be more. A simple example, a touch sensation in the face area usually is more. When you touch the face, you feel the touch much better than the touch which is felt at the buttocks or back. The number of sensory nerve fibers at that area would be lesser in the buttocks and back comparatively, whereas compared to the face area, and hence the face area is considered to be more sensitive to the touch. Similarly, these different type of sensation carrying nerve fibers or nerve endings, the tactile hair nerve endings, they have the sensation of 2-point discrimination. They give that 2-point discrimination sensation. Then end organ, refinous end organ also helps in the same pain and touch sensations. It helps in that pain and touch sensation. Golgi tendon apparatus is in the muscles and the tone of the muscles. The strength, stretch on the muscles is felt by Golgi tendon or muscle spindles. More of these we will be discussing more in detail when we go into the muscle diseases, etc. And crassus corpuscle and mesenus corpuscle also carry the 2-point discrimination, pain and touch sensation. So they get stimulus as such. Now all that important is whether a system has capacity to carry that sensation or not would be deciding, like whether that organ of the body or that part of the body can receive the sensation or not. That's why the eyes only can receive the light, and the ears only can receive the sound, the skin only can receive the touch sensation, and so on and so forth. So that 5 sensory organs, Pancha Indriyas, they are located at the specified areas, and each of them have a molecule of the specified Panchamahabhuta, that theory, that the same can be attributed to these modified nerve endings, and which can respond to only a specific type of the stimulus. So ultimately, any type of the stimulus would be converted into an electric energy, and that electric energy would be passing through the nerve fibers, which produces the functions. That's exactly the vata which gets stimulated. Now that's one part. Now once the impulses reach to the muscles, now what happens in the muscles? Nerves, when they cut the synapse, there will be a synapse at the muscle area also. And if the impulses reach to the muscles, nerves connected to the muscles, that's what we call as the neuromuscular junction, which also is a modified synapse. There again, there would be the neurotransmitter secreted mostly in the skeletal muscles. It's acetylcholine, and that acetylcholine secreted would be producing an action potential in the muscles. In the muscles, you have the structures which we call as the actin and myosin fibrils, which can slide over. So once there is a stimulus, there will be in the nerves which are connected to the muscles, which we call as motor nerves. See, when the conduction of the impulses reaches from a higher center to the periphery, or reaches to the muscles which produce an activity, that's called as the motor nerves. Now, in the muscles, you have these actin and myosin fibrils, which are located in a form of a spring. So once there is a stimulus, there will be a slight overriding, and hence the muscle becomes shortened or it becomes contracted, as you see in this picture. Like there could be initially the gap between these spindles would be more, these actin and myosin fibers will be more. Once there is a stimulus, it gets to get contracted and it gets to close together, and that is shorter. It requires a series of chemical activity. Of course, more of that chemical activity we will discuss later on. But if there is a single stimulus, this reaction is only momentary, temporary. Immediately it gets relaxed, or the muscle comes back to the previous position. And to keep that muscles in a continuous contraction, there has to be a continuous impulses. If there is a continuous impulses reaching to the muscle tendon, then these impulses would be summating together, and you will come to a stage where the muscles remain contracted, and this stage is called as tetanic contraction. So muscle tension, single stimulus, if there is a single stimulus from the nerves, then there could be a twitching, a contraction of the muscle, and then again the muscle can be relaxed. If there is a continuous impulses at a specified rate, it reaches to a specified rate, then the muscles can contract smoothly and the muscle can remain contracted. All our physiological activities, they need a tetanic contraction, and that tetanic contraction is achieved by having a continuous impulse of stimulus to the muscles. Now, all that variable factors which we have decided, we discussed, like the speed, different nerves with the different speed of conduction, nerves with the myelination or non-myelination, nerves with the having that different capability of changing their excitability, like or that membrane, the summation, membrane potential variations, all these come into the picture in maintaining that tetanic contraction. When we go into the diseases of the nerves and presenting with that different activities of voluntary and involuntary activities of the muscles, the importance of these servo mechanisms, these we call all that put together, that all that mechanisms which keep the muscles contracted as a servo mechanism. Or maybe in a simpler version, suppose you have to make a muscle contracted, like if you have to pick an object, consciously you will be picking the object with a conscious effort. But once you have held the object, you can just forget about that, but still your fingers can hold on to the object till again you consciously stop that activity and relax the muscles. Suppose we are picking up a pen, you have picked up the pen with a conscious effort, but once you have picked up the pen, then even if your attention is towards the other side, the pen will remain in the hand, that's your muscles will remain contracted in a tetanic contraction, and the pen will be held as such. And this kind of a mechanism can be maintained when there is a continuous volley of impulses from different areas, which is generated by a single impulse. See, a single impulse is presented by your conscious effort, but in the subconscious level, there has to be continuous impulses generated to keep the muscles, and this kind of a mechanism is possible because of those variable factors which we have discussed, like the excitatory potentials and these membrane potentials, differences as such, so on and so forth. And more of that specific details we will be discussing when we go into the diseases there. And in some, all these processes are called as servo mechanisms. Servo mechanisms are the backup mechanisms which help in maintaining the contraction. This phenomena is to be clearly understood to know the certain of the diseases. Like that's one aspect. Now, the sensations are carried from the surface of the body through the spinal nerves, somatic nerves, mainly through the spinal cord, and the distribution of these sensory nerve endings at the different zones will be related to the specific spinal section, and this is what we call as the dermatomes. Suppose a cutaneous sensation, senses from the skin, the sensory nerve endings which are presented in the skin, that nerve endings are connected to the nerves and they reach to the spinal cord at a specified segment, and the surface area of the body related to that specified spinal segment is called as the dermatome, and that dermatome are presented in the same manner. Now, this is how the whole dermatomes are mapped on the body, and the anterior and the posterior surface. The upper surface of the body, head area, is by the cervical segments, whereas the lower area is by the sacral segments in between the thoracic segments, and this diagram helps you to locate the lesion if at all there is a pathology of sensation. The location of the lesion is possible, and more detail about that, how to locate that, would be discussed later on. Now, sensations which are carried through the spinal cord would be reaching to the a specified conducting cords, or which are called as a pathways. The different pathways are there through which it will be reaching to the hair center. Now, at the level of the spinal cord itself, some of the sensations are carried through the same side, and some may be crossing to the other side, and the sensations will be reaching to the brain area. More detail about that, like which of the segments, which tracts, as these are also called as spinal tracts. We will have more discussion when we go into the specified disease condition. Now, when we are discussing about the general map as such, this is about the general arrangement of the nerve fibers. And when it reaches to the cortex, in the cortex, each of these represent a body of the area is represented at the sensory cortex area, and this is how the usual diagram, like in which zone of the brain, which part of the body is sensitized. What I mean is, suppose you have touched the hands as such, then the stimulus would be reaching to the brain through the nerve tract, and it will be ending up the area where the hand is represented. And if the stimulus reaches to that area, then you know that your hand is touched, and there is a sensory stimulus from the nerves as such. Then once the sensory stimulus reaches to the nerves, it is felt or received by the specified areas of the brain, and the specified areas of the brain are again categorized based upon their functions. Like the primary sensory area is the area where it is received, and then there will be sensory association areas, vernix area, auditory and visual area. Now then mental functions also are there, and there is a motor area which conducts or which gives an impulse to the periphery to produce the reaction. Now the other areas, they help in planning that what is the required for the reaction. So if there is a stimulus, then what should be the reaction? This is done by these different areas. At this moment, I will not go into the details of these functions, like which are those areas which are nuclei regulating this. We will be discussing those nuclei and their functions as we go into the disease conditions. So from the gross point of view of organization of the nervous system, I would simply say that if there is a sensory stimulus, all these areas would help in integrating that sensation into a meaningful motor activity. Like if there is some touch sensation, if somebody has touched your body, what next has to be done? This is thought of, and then that reaction is sent through the stimulus which is responsible for that reaction is sent through the motor area, the primary motor function area. The primary motor function area of the brain is in the midbrain towards the anterior side, and there again the different areas of the body are represented in the same. Like if you have to shake the hand, if there is somebody touches and then you have to shake the hand, the stimulus would be reaching to that from the hand area to the nerve fibers, which reach to the muscles of the hand, and that would be producing the contraction as such. These stimulus again would be carried through from the cortex through a tract, descending tract. We call these tracts as the descending tracts, and there again some of the nerve fibers would be crossing to the other side, and some of the nerve fibers will not cross to the other side, and this is what we call as a pyramid formation. And you will have a extra pyramidal and pyramidal tracts. The tracts of nerve fibers which cross to the other side, they are called as the pyramidal tracts, and those which do not cross to the other side, they are called as the extra pyramidal tracts. Importance of them, what is their functions, and detail of that we will be discussing more when we go further deep into the subject. So in the present, in the planning, general planning, I will leave it to that part, like it reaches to that. And of course the impulses, though it's generated in the cortex, there will be some more meaningful input into that impulse, modification of the impulses at the different areas, which are those modifications of the cerebellum and substantia nigra and so on. We will come to that later on. We will come to that part later on, like what the cerebellum does, what the thalamus does. We will come to that part later on. But all of them, they make this activity meaningful. For shaking hand, you need to have lots of complex activity. The muscles have to be contracted, and the muscles have to be contracted at the level which is required. As such, it's not that they should be contracted maximum. There has to be coordination of many organs of the body. It's not only the hands which have to move. There has to be a expression through your cranial nerves, your eyes, your mouth also have to react to that. So the whole activity has to be meaningful, and all these would be adding to that. Which component adds to which area, which part, that we will be discussing when we go into the later in the disease portion as such. So this integration is the main function of the brain, and then the impulse reaches to the muscle at the periphery, and the muscle will be contracted. The impulses which are crossing through the spinal cord, they are named as, these areas are called as tracts, and these in the spinal cord you will have few ascending tracts and few descending tracts. Motor tracts are descending tracts and sensory tracts are ascending tracts, and they are named depending upon the target to which they are reaching or the origin from where they are occurring. Like these spinocerebellar tracts are the tract or the area where the nerve fibers are connecting from the periphery spine to the cerebellum, and the spinothalamic tracts are which carry to the thalamus, and so on. Now, what are those functions of each of the tract? We will be discussing in detail later on. So, for the present, when we go into the organization, it's only that in the vertebral spinal cord, you will have these different tracts, ascending or descending tracts. The blue colors, they are the ascending tracts, red are the descending tracts shown in the picture, and they are named based upon the origin or the end point, end area of these tracts as such. And these are, so rubrospinal, reticulospinal, or oliospinal, vesiculospinal, in the sense they are from the these rubrous or red nucleus area, reticular area, and originating from these different areas as such. So that's how these are organized, and all these get their role to play when there is a meaningful activity as such. So now again, these meaningful activity, or that speed of conduction and the connect these servo mechanism could be affected, because you will have many types of the nerve fibers in between which can vary the speed of conduction. See, the corticospinal tracts, which are from the cortex, they are the fastest alpha motor neurons. Then the gamma motor neurons are mainly from the subthalamic tracts, like from the cerebellospinal tracts, so on. Now, but because of that, one impulse generated from the cortex can continuously produce a series of volley of impulses, battery of impulses, because of these different types of the nerve fibers which reach to the muscles, and the muscle can get activated and can produce a meaningful contraction, tetanic contraction as such. So that's how it's organized as such. Now, which more detailed factors of each of these we will discuss later on. Now, meaningful activity is produced due to a stimulus reaching to the higher center, and then there has to be integration, and a meaningful activity has to be performed. But the life needs some more mechanism. It's not that every time, if you have to have that kind of a time, like when there is a stimulus, you have to study the stimulus and then produce a reaction. The time taken could be longer, and it can affect the body, and hence the body has another mechanism, supportive mechanism of producing a reaction or another level of integration of the nervous system, which is called as the local reactions or reflex actions. There could be a reflex action. Reflex action is, if there is a stimulus at one end, then the stimulus would reach to the higher center, and that higher center will take its role later on. But immediately some of those impulses will be connected to, or it will be reaching to the motor neuron at that level through short segments, again by the excitatory or inhibitory. At times it could be excitatory and inhibitory, and this will produce a immediate action of the muscles related to that area, where there would be an immediate response, and usually this response is of an extension, like it will be moving away from the source of stimulus. If you have suppose if somebody touches with a surprise, like without you are aware of this, immediately you will have a jerky movement, and that jerky movement is produced due to the reflex action, where your thought process is not involved. You have not become conscious of the factor that you are touched. At that level, the reflex action would occur, and this reflex action is again subdued. It's not allowed to go into a maximum limit, and it is reduced, subdued by the other mechanisms which are by the higher centers. Now this also is another area. So there is a moderate integration at the spinal cord level too. So usually integration is a maximum in the brain, the different areas, but a moderate integration would occur at the spinal cord level too, and hence even that reflex activity is somewhat moderated, so that it doesn't go to that level where it can harm. And variations of this reflex activity can represent some of the diseases as such. So this way the whole organ is whole nervous system is organized, and for the sake of convenience we consider the brain and the spinal cord as the central nervous system, because they are located in the midline, and the others as the peripheral nervous system. That naming of peripheral and central nervous system is only from the point of view of this area, rather the location. And of course there is a moderate level of integration in the central nervous system. There could be an integration, a moderate integration at the spinal level, and a high intensity integration at the brain level as such. This is how the whole activity is regulated. Now further, it's not only that motor and sensory activity, the same activity is converted into different levels as such, and ultimately it results in what we call as a mental capacity or intellect. And this again would be of different schemes as such. The basic of the central nervous system would be sensations, receiving the sensation, and producing an activity as such. The sensations could be olfactory, visual, auditory, or the movement as such. Then another factor would be, because of these sensations you can have a meaningful activity, you can have a stable position, postural security, that you can stand erect or may lie down or support yourselves properly, and you are aware of your sides of the body, and you can plan your activity, like suppose if there is a stimulus or some painful stimulus, you can move away from the painful stimulus. If you have to walk, you can walk, and so on. If you feel hungry, you can eat, and so on. Then the same would be further modified into an ability to screen and decide like whether this is good for you or whether it is not good for you, and you can plan your activity beyond that immediate stimulus for the next day also. Then it also helps in the further as you learn. Now these are all the process of learning or evolution of the functions. In the next level, eye hand coordination, like whatever you see and you hold that object, that again needs a coordination between the eye and so on, the movements of the eyes, so, and adjustment of the position depending upon the requirement, like when you are pushed you can correct yourself. This kind of skill is developed next. So this is how the whole process of learning is developed. A newborn may have only this basic sensation, but later on as it occurs, development occurs, it comes to that level, and ultimately it results in an intellect, a capacity to memorize, capacity to recall the memory, capacity to renew or have a fresh accept this ability to think and then decide about that. So that's how the whole functions are evolved. So the whole functions are very complex functions, and hence the study of the neurology is considered to be a complex subject, difficult subject. But when we go into piece by piece, each of these, it doesn't become so complex. It is a simple as such. Now, as far as the other aspects, some more basic aspects of the nervous system, again I will not go into all the detail, very basic aspects. The total number of nerve cells which we have, including in the brain and so on, is much more than what's really needed, and that's what we call as the reserve. And it is calculated that the reserve is about 1:300. So at the most we will be using only one nerve cell out of the 300 to learn this and to reach that level of intellectual capacity. 299 nerve cells would be kept as reserve. So, and these reserve nerve cells help in recuperating or recovery from a damage to the nerve cells. But at the same time, the nerve cells also can regenerate, particularly peripheral nerve cells can regenerate if there is an axonal damage. But if the neurons are damaged, then they may not regenerate. Till very recently, it was believed that the central nervous system cells cannot regenerate, but very recently there are some new inventions which have suggested that central nervous system cells also can regenerate. So whether a person can recover from an injury to the nerve or not is depending upon one, the capacity of that particular nerve cells to regenerate. Axonal regeneration, if the damage is to the axons, it may regenerate by axonal regeneration or remyelination. Or the other way can be the other excess of the nerve fibers, whether they can take the role or not, that depends upon their anatomical location. And of course, whether the real cells can regenerate or not is a controversial issue. Majority of the people would believe that the nerves cannot regenerate. Central nervous system cells cannot regenerate, but some new researchers have come up which show that it can be regenerated. Now, another aspect of the physiology of the nerves is that nerve cells are depending upon the nourishment continuously, and particularly oxygen is required continuously, and they do not have a capacity to switch over to anaerobic metabolism. And hence, if the oxygen supply to the nerve is cut off, even by a short duration, the nerves can become dead, they can be affected, and the average time duration in which the nerves can get damaged in an environment of anoxia, where there is no oxygen, would be around 5 minutes. Beyond that, they may not survive on an average, because they do not have a capacity to switch over to anaerobic metabolism. And continuous blood supply is required. But the blood supply to the nerves is not the same, similar as that of the other tissues. Here the blood cannot come into contact with the nerves directly. There is a continuous layer which is called as the junctions layer. Endothelial cells of the capillaries, they have a tight junction where the cells will not migrate, only the endolymph will be migrating, reaching to the nerves, and the nerves will take the nourishment from the endolymph, from the blood which has leaked through, or the fluid which leaks through. So, and hence, all the substances, chemicals in the nerves, in the blood cannot reach to the nerves, and only specified substances can reach to the nerves as such. And this is actually based for the protection of the nerves, because the nerves are highly sensitive and highly sophisticated. They are more sensitive to the damage. To avoid a possible damage, the nature has created that blood-brain barrier, but it also has an importance from the pharmacological point of view. Many of the drugs may not reach to the nerves easily, and only those drugs which can cross this blood-brain barrier can reach to the nerves, including the subject which you study. With this, we will conclude today. In the next, we will discuss about the basics of examination, neurological examination. If there are any questions, we will try to answer, and then, right, there are no questions, so we will conclude today. We will continue in the next session about neurological examination.